Viruses are a unique group of pathogens with a simple acellular organization and a distinct pattern of multiplication. Despite their simple structure, they are a major cause of disease. They have no cytoplasmic membrane, cytosol, or functional organelles, but they can infect all types of cells, and numerous viruses can also infect bacteria, which are called bacteriophages. Viruses and bacteriophages are not capable of metabolic activity on their own, so instead, they invade other cells and use their metabolic machinery to produce more viral molecules, nucleic acid, and proteins, which then assemble into new viruses. Viruses can exist either extracellularly or intracellularly. In the extracellular state, the virus is called a virion and isn't capable of reproducing. A virion consists of a protein coat called a capsid surrounding a nucleic acid core which contains the genetic material or the viral genome. The nucleic acid and the capsid are collectively called a nucleocapsid. Some virions have a phospholipid membrane derived from the host cell called an envelope, which surrounds the nucleocapsid. The viruses that have an envelope are called enveloped viruses, and these include the herpes viruses and HIV, while the ones that lack the envelope, such as poliovirus, are called non-enveloped or naked viruses. Once inside the cell, the virus enters the intracellular state, where the capsid is removed and the virus becomes active. In this state, the virus exists solely as nucleic acids that induce the host to synthesize viral components from which virions are assembled and eventually released. Now the viruses are surrounded by an outer protein coating called a capsid, which protects the viral genome and aids in its transfer between host cells. Also, according to their capsid symmetry, the viruses can come in many shapes and sizes. There are 3 types of shapes: helical, icosahedral, and complex. First, the helical viruses have a capsid with a central cavity or a hollow tube, which is made by proteins arranged in a circular fashion, creating a disc-like shape. The disc shapes are attached helically, creating a tube with room for the nucleic acid in the middle. An example of a virus with helical symmetry is the tobacco mosaic virus, which is the most studied example. Moving on to the icosahedral viruses, which are made up of equilateral triangles fused together in a spherical shape that fully encloses the genetic material. These viruses are released into the environment when the cell dies, breaks down, and lyses, thus releasing the virions. Some examples of icosahedral viruses include poliovirus, rhinovirus, and adenovirus. Finally, the complex viruses have a combination of icosahedral and helical shape, and may have a complex outer wall or head-tail morphology. The head-tail morphology is unique to viruses that infect bacteria, called bacteriophages, and they have an icosahedral-shaped head and a helical-shaped tail. These viruses use their tail to attach to the bacterium, and then they create a hole in the cell wall and insert their DNA into the cell using the tail as a channel. Now, the nucleic acid core of the virus contains the genetic material, which can either be DNA or RNA, and the total genetic content of the virus is called the viral genome. The viral genomes are usually small and contain only a few genes that encode for proteins needed by the virus that's not present in the host cell. The genetic material can be single-stranded or double-stranded, linear or circular. In DNA viruses, the viral DNA enters the nucleus and uses the host's DNA polymerase to create more copies of itself, as well as viral mRNA that gets translated into viral protein. With RNA viruses, many of them stay in the cytoplasm, where the viral RNA is used by the host's ribosomes to create viral RNA polymerase proteins. These will help replicate the viral genome so more viruses can be created. RNA polymerases are more likely to make copying errors than DNA polymerases, and therefore often make mistakes during transcription. This is why mutations in RNA viruses occur more frequently than in DNA viruses. Alright, as a quick recap, viruses are a unique group of infectious agents with a simple acellular organization and a distinct pattern of replication. They can exist either extracellularly, where the virus is inactive and isn't capable of reproducing, or intracellularly, where the capsid is removed and the virus becomes active. A virus in the extracellular state is called a virion and consists of a protein coat called a capsid, which protects the viral genome and aids in its transfer between host cells. The capsid is surrounding a nucleic acid core, which contains the genetic material or the viral genome, which can be either DNA or RNA. Some virions have a phospholipid membrane derived from the host cell called an envelope, which surrounds the nucleocapsid. According to their capsid symmetry, the viruses can come in many shapes and sizes, and they can have 3 types of shapes: helical, icosahedral, and complex.
Influenza is one of those perfectly infectious viruses. It's transmitted in microdroplets and aerosolized when we sneeze and cough. It gets on our hands and we touch things, and it can live a little bit longer in the environment than many viruses can.
You go in a subway, somebody sneezes on you, somebody blows their nose, they shake your hand. So from the standpoint of spread, it's of great concern. Some people who get infected can wind up getting serious complications, such as viral pneumonia. And then what happens when you get very ill, you get multi-system failure.
Year in and year out, influenza kills tens of thousands of people in the United States alone. It infects millions upon millions of people. It shoots around the planet with new strains coming up all the time.
Flu can come from anywhere that humans are in daily direct contact with animal populations. And every nation on Earth has animal farms, poultry farms, swine farms. We are lucky on a day-to-day basis. I think the potential for flu to turn into something more deadly is always there.
But here's the thing with influenza that is particularly concerning. These wild waterfowl viruses have the ability to jump out of their wild bird reservoirs and get into other unrelated hosts.
So some of them are predominantly in poultry. Some of those are also in other animals like pigs. We have swine flu. We have some of them in horses. We have equine flu. They can be in dogs. You can have canine flu and so on.
And it's this ability of viruses to move around and stably adapt to different hosts that gives influenza its greatest sort of biological advantage, but makes it the most difficult to control.
They're jumping from one species to the next, and there's a lot happening. They're mutating, they're recombining, they're exchanging their little genetic segments.
And they create brand new influenza viruses never seen before.
It's in that process of birds to other animal species to humans, and then humans to humans, that flu has become such a dangerous virus to humans. That's what makes, in part, influenza the king of infectious diseases. Highly efficient at transmission and highly efficient as a killer. You put those two together, that's the ultimate fire and gas combination.
When you have factory farming, you could have millions of chickens or millions of ducks all in very close confined areas.
Flu likes crowds. Flu likes close contact. So A virus getting into a massive poultry farm is fantastic for that virus.
What you don't want to happen, and it hasn't happened yet with those bird flus, is that it jumps species and then it adapts itself to rapidly and readily and efficiently spread from human to human.
But there's no reason to believe that a virus like that won't figure out a way, evolutionarily, to infect us really easily. And then a lot of people are gonna die unless we do something.
Every cell in your body is a tiny factory, constantly making proteins. Your genetic code, or DNA, directs every activity. Protein-making machinery in the cell reads the cell's own DNA and makes sure the cell proteins go where they're needed. Viruses have their own genetic code, but they don't have the machinery to copy their DNA. So viruses need your cell's machinery to reproduce themselves. A virus starts this process by using the keys on its surface to gain entry. Like a Trojan horse, it enters the cell. The virus releases its own DNA into the unsuspecting cell and hijacks the protein-making machinery. Now, this vital cell function becomes an assembly line for new virus particles. No longer in service to your body, the cell now serves the virus, manufacturing thousands of new virus particles, which are released to enslave more cells, thousands and thousands more.
Okay, so the topic of this video will be viruses and the lytic cycle. So let's go ahead and get started. So first of all, let's begin with what is a virus. You know, when you look at the definition of a virus, it's pretty vague. A biological particle composed of nucleic acid and protein. Well, one of the more commonly known facts of a virus is that they're parasites, but they're a little different than parasites that we typically think of, such as a tapeworm. Viruses are intracellular parasites, meaning that they actually reside inside of a cell of a host. And that's what this video is going to focus on in just a little bit. Well, when we look at this diagram, we see the 6 kingdoms of life. Plants, animals, fungus, protista, and then there's 2 bacteria kingdoms. So if something is alive on Earth, it's classified in one of these 6 kingdoms here. So where do viruses fit in? You know, the answer is nowhere. Viruses are not classified as a living organism. Well, let's focus on that a little more right now. What I want to look at is, you know, some of the characteristics that viruses lack that really prevent them from being classified as a living organism, yet they actually do possess some characteristics in common with life. Well, let's go ahead and look at the no category on this table here. Viruses are not considered alive for a few reasons. Number one, they're not made of cells or organelles. They cannot reproduce on their own without a host. So here's a cell going through mitosis. You know, viruses don't do this. Viruses do not metabolize energy. What that means is they're like this caterpillar here, they're not herbivores. Like these wolves hunting, they're not carnivores. Like this algae here, they're not autotrophs. They don't feed, they don't take in energy. And they don't really perform any cellular processes on their own. So the molecule of ATP right here should remind you of the process called cellular respiration. Well, viruses don't produce ATP and they don't perform cellular respiration. Viruses do not do photosynthesis. Viruses do not do DNA replication. However, replication will be an important topic in a little bit. Viruses themselves do not do transcription. Viruses themselves do not do translation. They don't possess the organelles. They don't possess the enzymes. They don't possess the machinery in order to do these processes. They don't actively transport molecules into and out of themselves. So what about the other side? Viruses do actually possess a few characteristics in common with life. For instance, viruses are able to replicate. They're able to make more of themselves. We're going to talk about the lytic cycle in just a moment. Viruses have nucleic acid. Viruses will contain either DNA or RNA as their genetic material, but they do possess nucleic acid, as does every other form of life on Earth. Viruses are able to adapt to their surroundings. The picture here shows some vials of AZT, which is an HIV medication. And HIV is actually, the virus HIV is adapting to the AZT medication, and therefore it's not working. The medication is not as effective as it once was because the HIV virus has adapted. Viruses have organization to them. They have parts that do functions, and we'll get into some of these in just a moment. So when we look at the basic structure of a virus, I want to mention that the shape and the size of viruses differ from one another. Now, they all do possess a few characteristics in common. For instance, you look at the three drawings here, and in each you'll see that the three viruses all have what's called a capsid, an outer coating, an outer layer made up of protein. And like the notes say, the protein coat or the capsid surrounds nucleic acid. Well, that tells me the second thing that all viruses have, which is nucleic acid. Now, the three pictures show the viruses with DNA inside, and some viruses possess RNA, so it really does depend upon the virus. The virus on the left, labeled bacteriophage, has what almost looks like creepy crawly legs on it for walking and crawling around. These are called tail fibers, and that's not what their function is. These tail fibers are used for anchoring and attaching onto their host cell. So do they actually look, do these bacteriophages actually look like this? The answer is yes, they actually do. Now, not all viruses look like this bacteriophage right here, but some of them you can see are kind of unique looking. So let's start the transition into how viruses actually replicate. Here's a picture of a bacteriophage, and just mentioned these a moment ago. And bacteriophages are viruses, but what's unique about them is they actually infect bacteria. So it's kind of interesting that bacteria can even be infected with viruses. Now, we often will study bacteriophages because how they replicate is fairly similar to how animal viruses that may get us sick, for instance, also replicate. So we can learn a lot and apply it to human use. Now, when it comes to viral replication, there's really two cycles, two replication pathways. One is called the lytic cycle and one is called the lysogenic cycle. The rest of this video is really going to focus on the lytic cycle. I have a different video if you'd like to learn more about the lysogenic cycle. So let's go ahead and dive into the lytic cycle. Well, we can break the lytic cycle into some stages, and the first one is called attachment. Like the name implies, the virus will attach and land onto a host, the cell membrane of a host. And the picture shows, here's our host cell, and here comes a virus that has just attached. Notice how there are these little objects on the outside of the cell called receptors. And what the receptor will do is actually kind of catch the virus. And so the virus has these little bumps on it made of protein, and if the virus matches the receptor, then attachment will be successful. It's kind of like the lock and key analogy here. The virus acts as a key, the receptor acts as a lock, and if the virus is able to match the receptor, the cell, not knowing anything is wrong, will draw the virus into itself. And that'll take us to step 2. Step 2 is called entry, and like the name implies, the virus will enter the host cell. The DNA or the RNA, the nucleic acid, will enter the host. Now there's a couple ways that this might happen. In some situations, the virus will literally inject the DNA or RNA into a host cell. That's what bacteriophages do. Another way that the nucleic acid of the virus can enter the cell is the entire virus will ultimately be swallowed by the cell, and that's what this animation is going to follow. Notice how a little pocket has begun to form. The cell is actively swallowing the virus and taking it into itself. As the process continues, notice how the virus is drawn deeper into the cell. And ultimately, there you go. The cell has swallowed up the virus and taken it inside of itself. Now what happens is enzymes from the virus will actually dissolve the outer capsid, the outer protein coat, thus leaving behind the nucleic acid of the virus. So this will now bring us to stage 3, called replication. And so what happens is the virus DNA that you see will perform the process called DNA replication to make more viral DNA. And that happens by the viral DNA entering the nucleus of the host cell. And now we're going to see the process of DNA replication, the process where DNA is used to make copies of more DNA. The reason the virus needs to go into the nucleus is because that's where the enzyme DNA polymerase is found. And so this enzyme DNA polymerase will direct DNA replication. Notice what's happening. More DNA of the virus is being copied and created. Well, what happens next is the virus DNA, and in just a moment, the RNA that it will create will be used to make proteins that belong to the virus. Well, how does this occur? This occurs by the process of transcription and translation. If you've forgotten what transcription is the process of creating RNA. Inside the nucleus, there's an enzyme called RNA polymerase that will direct the process of transcription. Notice what just happened. A single strand of RNA was just created and sent to a ribosome. Now that the RNA of the virus is at a ribosome, the process called translation can begin. Translation is the process of creating proteins, the difference being the cell will actually be creating proteins that belong to the virus. This translation process is performed by ribosomes. That's the job of a ribosome, to build proteins. The ribosome doesn't really realize that it's building proteins for a virus. So the ribosome will read the viral RNA and begin to build proteins that belong to the virus. And that's what the capsid of a virus is made from. The capsid of a virus is made from proteins. So the cell's ribosomes are actually building parts of future viruses. And now we come to the 4th stage called assembly. What happens is the new viruses that have been created start to self-assemble. Notice how the DNA comes together with the capsid. And when this occurs, now we have brand new viruses that were just assembled inside the cytoplasm of the host. And so now we come to the final stage called release. And what happens is focus on the ribosome that you see in the picture. Remember, the ribosome is building proteins that belong to the virus. Another protein that the ribosomes create that belongs to the virus are enzymes. And I've kind of drawn the enzyme to be in the shape of scissors to illustrate what these enzymes will do to the cell. So here's the ribosome making enzymes. Enzymes that are ultimately going to cause the host cell to lyse or to burst. And this is where the name lytic comes from. It comes from the word lyse, which means to burst. So these enzymes that were just created are going to pop little holes in the cell membrane. And ultimately, that's going to cause the cell to burst and the viruses to be released. Now, once the viruses are released, the cycle is simply going to repeat itself. The viruses are going to spread. Let's go follow one of those viruses. So the viruses have spread, and now notice what's happened. One of the new viruses that just created has found another host to infect to repeat the cycle. And so that's kind of the trickery of the lytic cycle right there, is that even though the virus doesn't possess any of the machinery to replicate and make more of itself, it will actually hijack and take over a cell which does possess the machinery to replicate the virus. So there you go. I hope this video was helpful. If you're in my biology class, pause the video, try to answer these questions on a separate sheet of paper. I'd be happy to check your answers before class or after class. Thanks for watching. Okay, so this video we're going to discuss viral replication following the lysogenic cycle. So let's go ahead and get started. Okay, so when we get into the steps of the lysogenic cycle, we can really break it down into 5 stages. And we're going to call the first one the attachment stage. And like the name implies, the virus will land on the cell membrane of the host. So here comes the virus. Notice how there are these little objects marked receptors on the cell membrane. The virus itself has these little bumps and little balls of protein on the outer surface. So the virus will actually attach to these receptors. You know, the lock and key analogy works really well here. The virus has these little bumps of proteins on them, and if they are able to match the receptor of the cell, then the attachment is successful. So when we move on into the second stage of the lysogenic cycle, that's called entry. Like the name implies, the nucleic acid, the genetic material, the DNA or the RNA of the virus is going to enter the host cell. This happens usually in one of 2 ways. One is that the nucleic acid is literally injected into the host cell. This animation, however, is going to follow the other way. And that is that the virus, the entire virus, is swallowed into the cell. Notice how an indentation has begun. More and more and more of the virus is being taken into the host. And eventually the entire virus has been taken into the host cell. So what happens next are enzymes inside the virus will cause the capsid to break down. This releases the nucleic acid into the cytoplasm of the host. Well, what actually happens next is that viral DNA will actually be spliced into and combine with the DNA of the host cell. So watch this. Enzymes will actually break open the host cell's genome, and that allows the viral DNA to bond with and fuse with the host cell DNA. This combination of part cell DNA in black and part virus DNA in purple is what is called a provirus. This occurs during the entry stage of the lysogenic cycle. So before we go any further, I want to zoom out for a wider view of our cell here. And what comes next is a latent time period. The provirus will enter a stage of latency. And the word latent simply means dormant. The viral DNA will lie dormant inside of the host cell, and the host cell carries on its activities as if nothing's wrong. And so the infected cell simply carries on and eventually divides by mitosis. The difference being every time the host cell divides, it makes a copy of the provirus. During the cell cycle of mitosis, the DNA is duplicated, and then the cell begins to divide into two. Notice how there are now two infected cells when originally there was only one. So each newly created cell contains a copy of the provirus. Now this process happens over and over and over and over, and might happen over and over and over for years, just depending upon the virus. So as the process happens over and over and over again, the cell on the left divides into two more infected cells. The cell on the right goes through mitosis and divides into two more infected cells for a total of 4 now. So the one infected cell became 2, 2 became 4, the 4 would continue on to 8, the 8 would continue on to 16, the 16 would continue on to 32, and so on and so on and so on. Well, now let's kind of get back to the lysogenic cycle here. Let's focus on one single cell, just for simplicity. And let's zoom on into that one cell for a closer look. So stage 3 is called replication. Eventually, the latent period in the stage of the lysogenic cycle will end. The viral DNA will become active, and this kind of signifies step 3 called replication. The DNA of the virus will perform DNA replication to make more DNA. And that's the definition of DNA replication, is the process of creating DNA from a DNA template. Remember, a template is kind of just another word for a mold. So in my animation, there's one molecule of DNA created, there's a second created, and there's a third. So multiple molecules of DNA are replicated. The DNA that was just created then undergoes transcription in order to make RNA. If you've forgotten what transcription is, it's the process of creating RNA from a DNA template. Now the difference being, the viral DNA in purple is going to be used to make viral RNA in purple as well. So there's one piece of viral RNA. Here's another molecule of viral RNA created, and here's another molecule of viral RNA created through the process called transcription. So now what happens next is the cell, the host cell, has ribosomes that will perform translation on behalf of the virus. Viruses don't have ribosomes themselves, so they kind of hijack ribosomes that belong to the host. So the definition of translation, if you've forgotten, is the process where a ribosome will create a protein. So there's a ribosome, there's a ribosome, there's a ribosome. And through the process of translation, a bunch of amino acids are gathered and collected at the ribosome to make a protein. And notice in this case, the protein looks an awful lot like the capsid of a new virus. That's because the capsid is made from of protein. So the same thing happens on the other piece of RNA. The process of translation occurs where the ribosome will actually gather a bunch of amino acids and build them together into a collection of protein. And there we see another capsid of another virus created. And one more time, here we have a ribosome collecting a bunch of amino acids, and as the amino acids are built together into a collection of protein, and here we have a third capsid created. So the process, keep in mind, we're now dealing with more than one infected cell because of that latent period when the cell went through mitosis and divided. The process also takes place inside of the other infected cells. And so we come to stage 4 called assembly. The viruses, the parts of the viruses that have been created will self-assemble, Here we see DNA and a capsid coming together to make a new virus. Here we see DNA and a capsid coming together to make a new virus. And finally, the third virus is created. And that leads us to step 5, known as release. Like the name implies, the viruses are going to be released from the host cell. And this happens in many cases because the virus will release enzymes that will pop little holes and poke little holes in the cell membrane of the host. This will actually cause the cell to lyse or to burst. Once that happens, those viruses are released to repeat the process. Hey, keep in mind, the process also takes place inside of the other infected body cells. So here are those 4 infected cells. Now I'm not implying that it's a ticking time bomb and all your cells are going to explode at one time. I'm not implying that at all. What I am trying to show you is that multiple cells will have their cell membranes punctured and multiple cells will burst. And this will release a large, massive amount of new viruses. And this is the lysogenic cycle. So as we finish up, pause the video if you want to try this practice quiz. And if you're in my biology class, bring me your answers before school or after school. I'd be happy to check your answers for accuracy. Good luck, everyone.
Vaccines and the immune response. How vaccines work. Influenza vaccines are able to trigger an immune response by mimicking viral infection. They are usually manufactured using inactivated or killed virus particles taken from various circulating influenza strains. Inactive fractioned viral components contain the subvirion particles hemagglutinin, HA, and neuraminidase, NA. In Canada, influenza vaccines are administered via intramuscular injection. These fractioned particles containing the foreign antigens hemagglutinin and neuraminidase are released from the vaccine into the bloodstream. There, they are met with an immune response mediated by various immune cells, including macrophages, T lymphocytes or T cells, and B lymphocytes or B cells. A major mechanism of action involves macrophage phagocytosis of hemagglutinin, breaking it into smaller components. After ingestion, macrophages display the hemagglutinin antigen on their surface in combination with a specific receptor known as the major histocompatibility complex, or MHC. T cells are now able to recognize and bind foreign antigens that are associated with the MHC. Upon binding to the MHC receptor, T cells become activated and proliferate into either cytotoxic T cells, regulatory suppressor T cells, or helper T cells. Activated helper T cells express hemagglutinin receptors specific to the vaccine strand on their surface and play a major role in antibody generation and memory B cell activation. Unlike T cells, B cells are able to ingest hemagglutinin independent of the MHC. Once internalized, B cells process the hemagglutinin antigen and present it on their surface in combination with an MHC. When activated helper T cells interact with activated B cells expressing antigen MHC receptors, they begin secreting lymphokines, which have several effects. Lymphokines trigger activated B cell proliferation, which leads to either their differentiation into memory B cells or into plasma cells. Plasma cells produce hemagglutinin antibodies specific to the strain of influenza contained in the vaccine. Memory B cells aid in future immune response when exposed to an active influenza virus. When an infected host sneezes towards an uninfected person, the nasopharynx is exposed to aerosol droplets containing whole, live influenza virus. Once inhaled, the influenza virus attempts viral colonization of nasopharyngeal epithelial cells. assuming that the vaccine strain matches that of the live virus. Hemagglutinin antibodies block viral attachment of live influenza virus to host epithelial cells, and overall disease is either avoided or diminished in severity.
If you'd been born in the US in the year 1900, when the three leading causes of death were all infectious diseases, pneumonia and flu, tuberculosis, and gastrointestinal infections, your life expectancy would have been just over 47 years. In the year 2010, the leading cause of death were chronic diseases like cancer and heart disease. We can thank improvements in sanitation practices and medical advances like vaccines for drastically reducing the impact of infectious diseases and extending our life expectancy to nearly 79 years. Before we learn exactly what a vaccine is and how they're made, let's discover how bacteria and viruses can make us sick in the first place. Both bacteria and viruses are microbes, tiny microorganisms that are too small to see with the naked eye. Viruses are smaller than bacteria. Most of the largest viruses are smaller than the tiniest bacteria. Both viruses and bacteria are spread by contact with infected people, animals, surfaces, or food, and both can cause diseases in humans that produce similar symptoms, like digestive upset, fever and chills, aches and pains, and respiratory distress. But despite those similarities, bacteria and viruses could not be more different. Bacteria are incredibly hardy living organisms. They can survive in nearly any environment and can reproduce on their own. Most bacteria are harmless to us, and many are actually helpful. Trillions of bacteria live all over our bodies, and scientists have recently discovered that our bodies might actually be only 43% human, and the rest of us made up of microscopic organisms like bacteria. Once a disease-causing bacteria has entered our system, it begins to reproduce and give off toxins, and it's these toxins that make us sick. Bacterial infections can be treated with antibiotics, which either kill the bacteria outright or limit its ability to grow. The bubonic plague, which killed 60% of the population of Europe in the 14th century, was caused by a bacteria. The Spanish flu epidemic of 1918, on the other hand, was caused by an aggressive strain of the influenza virus. A virus needs a host to survive and reproduce. Viruses make us ill by inserting their genetic material into our cells, causing those cells to burst and die or to mutate into malignant cells. It's nearly impossible to kill a virus once it finds a host. We can only treat the symptoms. The best thing we could do is try to prevent viral infections in the first place, and that's where vaccines come in. Now, before we can understand how vaccines work, we need to know a little bit more about our immune system. Our innate immune system's job is to monitor the body for pathogens and act as the first line of defense against infections. For the record, a pathogen is anything that can cause disease, like bacteria or viruses. An antigen is any substance capable of stimulating an immune response, which might only be part of a virus. Many organs and tissues are involved in our immune system. Our bone marrow is where the immune cells are born and constantly replenished. All immune cells start out as stem cells before developing into specific immune cells. Our lymph nodes are located strategically throughout our bodies, usually near openings where pathogens tend to enter, like our mouth or genital areas. Lymph nodes store immune cells to be deployed during an infection. The swollen nodes you feel when you're sick are the result of an increase in the number of cells in the tissue as your body ramps up its efforts to fight off the intruders. Our spleen filters pathogens from our blood as it circulates through our body, and our muscles contract to move air or liquids that contain pathogens out of the body by means of vomiting, diarrhea, coughing, or sneezing. Our skin acts as an important physical barrier against pathogens. The top layer of skin cells, called epithelial cells, line the openings of our body and are coated with mucus, a thick, sticky solution that makes it hard for pathogens to attach to our cells. The mucus coating also contains chemicals that help your skin maintain a slightly acidic pH, making it harder for viruses to survive. Some cells also have microfibers called cilia, which help move mucus and pathogens along the cell surface. Our nose hairs work similarly, filtering dust and other pathogens as we breathe. When a pathogen manages to sneak past our innate immune system, our adaptive immune system kicks in. The adaptive immune system reacts in specific ways to specific pathogens, whereas the innate immune system responds the same way to all threats. Antigen-presenting cells, or APCs, are produced in our bone marrow and monitor our bloodstreams for pathogens. When APCs encounter an unwanted intruder, they phagocytose it. breaking it into pieces and placing them on their surface before heading to the nearest lymph node to present the antigen signal and kickstart the full immune response. The signals from the APCs activate T cells, which oversee cytokine signaling. Cytokines are small proteins that fit perfectly with receptors on specific cells and give that cell instructions to help regulate the immune response. They may tell cells to grow, change, or reproduce, or instruct them to kill other cells infected by the virus. B cells are a type of immune cell that multiplies rapidly and secretes antibodies or immunoglobulins, or Ig, which are proteins that target and kill invading microbes. Once the infection has been dealt with, all that remains of your adaptive immune cells are a small number of T and B memory cells. After the primary immune response to the first exposure to the illness or to a vaccine, those lingering memory cells will make your secondary response to the same pathogen stronger and more effective, meaning future infections will be less severe, shorter, and may not present any symptoms at all. But now that we understand how our body fights off pathogens on its own, let's learn about how vaccines leverage our immune response to combat infections. So, what exactly is a vaccine? A vaccine is a compound that contains a killed or weakened version of a pathogen that is known to cause disease in humans. The vaccine won't make you sick, but it will stimulate an immune response. Unlike medicines that treat disease, vaccines work by preventing the infection in the first place. The immune response triggered by the vaccine produces the same memory cells that a true infection would, which teaches your body how to effectively deal with the pathogen in the future without the risks of having to get the disease first. Edward Jenner is credited with the first successful use of a modern vaccine in 1796 when he used cowpox material to inoculate a 13-year-old boy from smallpox. Louis Pasteur is another vaccine superstar credited with creating the first successful rabies vaccine in 1885. He's also famous for developing the process of pasteurization, which kills disease-causing microorganisms in food and beverages by heating them to a certain temperature. But the history of vaccines actually goes much farther back. Evidence suggests that the Chinese employed smallpox inoculation as early as the year 1000. Technically, this was variolation, not true inoculation, since their technique involved smearing open tears in a patient's skin with true cowpox, rather than a weakened version designed for vaccination. By the 1930s, vaccines and antitoxins were available for diphtheria, tetanus, anthrax, cholera, plague, typhoid, tuberculosis, and more. By the later half of the 20th century, thanks to the success of vaccines and vaccination programs, many previously deadly diseases had been all but eradicated in much of the world. Although the way vaccines work may seem simple in theory, creating a vaccine is actually quite complicated. First of all, the costs are astronomical. It can cost upwards of $1 billion and take more than 10 years to develop an effective new vaccine that we can be reasonably sure won't cause the disease. On top of that, viruses are constantly mutating, and we're constantly encountering new strains of the viruses like the flu. It's almost impossible to predict which strain will hit in a given year, and by the time an outbreak hits, there's usually not enough time to create a vaccine. For viruses that are more stable over time, there are many different methods for creating a vaccine. The method used depends on the pathogen being targeted, whether it's a virus or a bacteria, and the way it causes disease in our bodies. Live attenuated vaccines are created by weakening the virus so that it responds more slowly in our bodies, giving our immune system time to learn how to respond to the intruder without letting the disease run rampant. Normally, viruses replicate thousands of times, but attenuated viruses will only reproduce about 20 times, enough to stimulate the creation of memory cells without making you sick. The measles, mumps, and rubella, chickenpox, and some flu vaccines are all live attenuated vaccines. Live attenuated vaccines are made by passing the virus through a series of cell cultures or animal embryos up to 200 times. With each pass, the virus gets better and better at replicating in the culture or embryo, but worse at replicating in humans. The virus will still be recognizable to your immune system, but won't be able to replicate well enough in your body to cause disease. The advantage of live attenuated viruses is that 1 or 2 doses is usually sufficient to provide a lifelong immunity. However, these vaccines can't be given to people with weak immune systems. There's also a small risk that the virus will mutate and revert back to its original ability to replicate in humans. For example, the oral polio vaccine has a high risk of mutating and causing paralytic polio, which is why the oral vaccine is no longer used in the US. Inactivated vaccines are made by killing or deactivating the virus completely, so so that it can't reproduce at all, which means it can't cause disease. The polio, rabies, and hepatitis A vaccines are examples of inactivated vaccines. The pathogen is inactivated using heat or chemicals like formaldehyde, which destroys the virus's ability to replicate but leaves it recognizable to your immune system. Inactivated vaccines are advantageous because the inactivated virus cannot cause even a mild form of the disease, and these vaccines can be given to people with compromised immune systems. However, it usually takes several doses to achieve immunity, so additional booster shots will be needed. A subunit or conjugate vaccine involves removing one part of the virus and using it to provide immunity. These types of vaccines can be created in a few different ways. One method involves isolating a specific protein in the virus and presenting it on its own as an antigen. These virus-like particles, or VLPs, contain no genetic material and so can't cause illness, but still prompt an immune response which triggers memory cells and provides future protection from the virus. Yet another method uses genetic engineering to insert the genetic code from a disease-causing virus into another harmless virus to create what's called a recombinant vaccine. Your immune system will recognize the genetic coding of the virus, and memory cells will provide future immunity. Both types of vaccines can be used on people with weakened immune systems, and 1 or 2 doses usually provides lifetime immunity. The hepatitis B and human papillomavirus, or HPV vaccines, were both created through genetic engineering. But some diseases are caused by bacteria rather than viruses. Toxoid vaccines can be created to provide immunity against bacterial infections that are caused by the toxins released from the bacteria. Toxoid vaccines are created in a similar fashion to inactivated viral vaccines. The toxins are treated with heat or chemicals to create a toxoid, which is similar enough to produce an immune response, but cannot cause disease. For example, tetanus infections are caused by the Clostridium tetani bacteria, but the symptoms are actually caused by the neurotoxin tetanospasmin, which is released as the bacteria reproduces. The tetanus vaccine is a toxoid vaccine that teaches your immune system how to respond to the neurotoxin rather than to the bacteria itself. Passive immunity is created when you inject a person with antibodies, which immediately destroy pathogens. While you don't have to wait for your body to generate its own antibodies, like with most vaccines, the protection is usually only temporary. One of the most important parts of any vaccination strategy is the concept of herd immunity. When a significant portion of a population is vaccinated, it makes it difficult for a virus to travel from person to person, limiting its spread. Herd immunity is essential for protecting vulnerable people who can't be vaccinated, like very young children, older individuals, and people with compromised immune systems. These groups also tend to be the most at risk for complications from viral infections, so they're depending on herd immunity to protect them. Thanks to vaccines and the concept of herd immunity, smallpox has been completely eradicated. We've almost eliminated polio, many other diseases are held in check, and we're well on our way to creating vaccines for countless other viruses. Vaccines are truly one of the most important medical advances in history. If you enjoyed learning about how vaccines are made, be sure to check out our other videos like this one called What Made the Black Death So Deadly? Or perhaps you'll like this other video instead. As always, thanks for watching, and don't forget to like, share, and subscribe. See you next time.